Bismillahirrahmanirrahim. My name is Akram, and I'm going to present my research work entitled Comparative Study of B. propellus and B. pollen on Fish Growth, Biochemical and Hematological Indices of Grass Carp. I have completed my research work under the supervision of Dr. Farmanullah Dawad and Dr. Syed Ishtaq Anjum. This is my presentation outline: introduction, objective, material, method, result, conclusion, recommendation, and references. First of all, introduction. Bee products such as bee propolis and bee pollen are used as medicine from ancient time because of its biological properties as an antimicrobial, antiprotozoan, antifungal, and antiviral agents. Propolis is a sticky substance that is used by honeybees to maintain their structure of hives and softening the inner wall of hives and protect the entrance of foreign particles. Bee pollen is a natural compound that is composed of protein, essential amino acids, sugar, lipid, nucleic acid, and crude fibers. The composition of bee pollens vary due to differences in biogeographic origin, ecological habitat, and season. The previous study reported that bee pollen is used as dietary supplement for human and has antioxidant activity. Tenuferengodon edela, commonly known as grass carp, was imported from China to Pakistan in 1964 for biological aquatic weed controls in natural waterways. It shows as world's second largest aquaculture industry, which make about 14.7% of the global aquaculture production, which annually increase at average of 14% in China. Grass carp is a common and valuable freshwater species in China and other Asian countries due to its rapid growth, high meat quality, and taste. This study has the following objectives. First one, to analyze the effect of bee propolis and bee pollen on growth and biochemical indices of grass carp. Second one is to investigate the effect of bee propolis and bee pollen on hematological indices of grass carp. Material and method. First is collection and acclimatization of fishes. We collected fishes from Mughal Fish Hatchery, Punjab, and then transferred to the lab of Fisheries and Aquaculture Department of Zoology Cust for acclimatization. Fishes were acclimatized for 2 weeks to the lab condition. Then diet preparation. Diet was prepared from 2.5% of propolis and pollen incorporated with basal diet. After that, after the trial, first we take initial weight from the fishes, which we called initial measurement, morphometric measurement. And after the trial, after the end of the experiment, we again measure the weight, length, and we do the morphometric measurement of fishes. Biochemical analysis. For biochemical analysis, we take blood from the caudal vein of the fishes. And also for hematological analysis, we take blood from the caudal vein of the fishes. Growth performance was calculated according to the following formulas. Average daily gain is equal to average final weight minus average initial weight divided by feeding periods. We also measure weight, length, and girth of the fishes. Results. First of all, we measure the hematological parameters. In hematological parameter, MCV were measured. As compared to control group, the propolis and pollen shows significant increase in MCV values. MCH in MCH as compared to control group, propolis show average increase in MCH values, while pollen treated groups shows significant increase in MCH values. MCHC as compared to control group, propolis and pollen treated group show increase in treated groups. In RBC, as compared to control group, in pollen treated group, the level of RBC decrease WBC, white blood cells as compared to control group in pollen treated groups and propolis treated groups. They increase the level of WBC increase as compared to control group and all treated groups. Hematocrit value in propolis treated group and hematocrit value show average decrease, while in pollen treated groups it show no increase or decrease, no changes. After that, biochemical parameters. For biochemical parameter, we take blood from the caudal vein and then analyze the following parameter. The enzyme ALT, AST, LDH, and ALP were analyzed as compared to control group. In propolis treated group show significant decrease in these enzymes, while in pollen treated groups it show average decrease. The level of ALP, AST, LDH, and ALP, the decrease value in the body show healthy condition of fishes. After that, we do growth performance. In growth performance, we measure length, girth, average daily gain, and weight. As compared to control group, pollen treated and propolis treated group show a significant increase in the in these parameters: length, girth, average daily gain, and weight. Conclusion: honeybees products such as pollen and propolis are natural food ingredients, would be better and safer and helpful for the health and different parameters of fishes. Using concentration lower than 2.5% of bee pollen and propolis would be favorable for hematology and some biochemical parameter of grass carp. A recommendation: dose-dependent effects of propolis and pollen on different parameters of fish can be favorable, opening new perspectives of investigation on their biological properties and utilization. Also, further studies need to be undertaken to elaborate. I'm very thankful to Dr. Farmanullah Dawad, my supervisor, and Dr. Said Ishtiaq Anjum, my co-supervisor, and Dr. Shahid Niaz Khan, Mr. Muhammad Rauf Afridi, and Mr. Nurul Akbar sir, and all my research colleagues and family for their continued support in my whole research work. And thank you all. These are references. And thank you.